Hi everyone, welcome back to my YouTube channel for a new video in the recombinant DNA technology series. In this video, we'll talk about the different types of vector. However, in my next video, I'll elaborate individual type of vector. Now let us first talk about the definition of a vector. What is a vector? A vector is a self-replicating DNA molecule. So this indicates that this must be able to replicate independent of the host chromosome, and then it is attached to the foreign DNA fragment to be introduced into a cell which has features that make it easier to insert DNA. So that means that your insert must be accommodated into the vector. So that is the purpose of course. And select for presence of vector in the cell. So this is very much important. That means you must have an option in the vector so that you can select between recombinant and non-recombinant host or bacteria. Next we'll talk about the essential features of cloning vector. The number one feature is that the origin of replication, which means that the vector must have a sequence that will help it replicate inside the host cell. The second feature is the antibiotic resistance genes. So in other words, this is also called gene for selection. So using the specific antibiotic of which the resistance gene is present, we can select between recombinant and non-recombinant host cells, or in most of the cases the host cells are bacteria. So that means if you apply the specific antibiotic, the bacteria that harbors your construct, that will be able to survive, and the bacteria which do not harbor this specific vector will be killed. So that is the purpose of having antibiotic resistance genes. And then the third type is called the cloning sites. So cloning sites are also called multiple cloning sites or unique restriction sites. As the name indicates, you are able to understand that. So this specific portion will contain sequences onto which the restriction enzymes will be able to perform or cut or digest. So why they are called multiple cloning sites? Because there will be cut sites for different types of restriction enzymes, and they are also called unique restriction sites because each site is present only once. So in brief, you can say there are three essential features of a cloning vector. The one is origin of replication, the number two is antibiotic resistant genes, and the multiple cloning sites. The next question arises that why do you even need different types of cloning vector? So what factor determines which vector you need? I am going to describe them one by one. The number one you can see is the insert size. So whether the insert is very small in size or it's very large in size, so that will determine which vector you are going to use, because certain vectors like plasmids, they can only accommodate a smaller size of insert. However, if your insert is a bigger size or the genomic DNA, so then you cannot use plasmid because plasmid cannot accommodate a bigger size insert. In that case, you'll have to use some other vector, for example cosmid or yak or BAC. And next is the choice of host. Sometimes people use mammalian vector because they want to clone a mammalian gene, and sometimes people also use bacterial host. In that case, you'll have to use a bacterial vector. The next factor is that whether your gene of interest codes for whether the functional gene is consisted of single polypeptide or it has more than one polypeptide. So depending upon whether it consists of single or multiple polypeptides, you'll have to select your vector. Next is the gene product. If you are interested in protein expression from that particular gene that you are going to clone, then you'll have to be very cautious, because if you want to express mammalian protein, and that if you express in a bacterial system, that might give you many problems. So in that case, you'll have to select a mammalian vector and mammalian host. So in that way, it will determine what kind of vector you actually need. And finally, subcloning. So subcloning is a procedure that is defined by the fact that when you just want to clone from one vector to another, so that is what is called subcloning. For subcloning, there are multiple vectors available. So all these vectors together, they define which vector you need for your cloning experiment. So that is the reason multiple types of cloning vectors exist. And of course these vectors they don't exist naturally, so scientists make them in laboratory. So they synthesize, they perform various kind of manipulation or genetic engineering, and then they make that vector suitable for their use. Now I'll move to the types of cloning vector. So there are multiple types available. So the first and very kind of popular type is called plasmid. So these plasmids are actually genetically engineered plasmids of E. coli, and plasmids are very popularly used cloning vector in experimental biology. And the second type is called cosmids. So cosmids are lambda based vector, lambda bacteriophage vector. They are also called hybrid vector because they can replicate both in bacteriophage as well as in bacteria. So they consist of the cos site of the lambda genome plus the plasmid. Then you have phagemid. So phagemid, as the name suggests, you can understand it consists of the genome of bacteriophage plus plasmid. Sometimes people also use genetically engineered genome of lambda bacteriophage as vector. So that is called lambda bacteriophage vector. Then you have the genome of filamentous phage virus called M13. So the advantage of using M13 is single stranded. And now interestingly the sixth type is called BAC or BAC. BAC means bacterial artificial chromosome. So the chromosomes of bacteria, sometimes they are genetically engineered and manipulated in such a way that they can also serve as a vector. So yeast chromosomes can also serve as a high capacity vector after proper manipulation and genetically engineering, engineer. And also you have mammalian artificial chromosome or MAC. The final type is called expression vector. So as the name indicates, these vectors are for protein expression. So if you want to express a particular protein, in that case you have to choose an expression vector. VR322 is one of the first vectors to be developed in laboratory. Are you curious why the name is a very strange name like PBR322? Okay, that is because the P indicates that this is indeed a plasmid, and the BR identifies the laboratory in which the vector was originally constructed. So we know that BR stands for Bolivar and Rodrigue, the duo researcher scientist who developed the vector in the laboratory. And the 322, this number indicates the serial number of the plasmid. So this number particularly distinguishes this plasmid from others developed in the same laboratory. So that means in their laboratory they might have another plasmid which is called PBR323, 24, and so on and so forth. Now you can see the map of the PBR322 plasmid vector. So this map shows you the position of antibiotic resistance gene. Here you see the ampicillin resistant gene, and you see here the TET-R. So this gene codes for resistance against tetracycline antibiotic. Then you have a ORI sequence here. So this is for origin of replication, and then you have here the multiple restriction sites. Because this plasmid is very small in size, for example, this consists of 4363 base pair. So this is very much easy to handle in the laboratory. And because there are two antibiotic resistance genes, one is ampicillin resistance and another is tetracycline resistance, so this will give you added advantage while selecting your recombinant host. Also, this is a high copy number plasmid. Here the copy number indicates the number of plasmid per cell. So whether a vector will be high copy number or low copy number, it depends upon the ORI sequence. Overall, we understood that different types of gene vectors are available in the laboratory, and uses of these vectors are dependent on the downstream application. Thank you very much. See you in the next video.